Alyssa Koehler, our plant pathologist, is our next speaker. So, and it looks like she's ready. Go ahead, Alyssa.
Right. Good morning. So today I'll be talking to you a bit about soil-borne diseases within processing vegetables. Happy to be with you virtually today. Sorry I can't be in person, but just as a reminder, I am located at the Causal Research and Education Center. I primarily cover field crops, corn, soy, wheat, barley, but more and more find myself getting a little bit more involved in the vegetable scene. And today I'll be talking to you on this theme of soil-borne diseases in processing vegetables, mostly focusing within lima and snap beans, a little bit on pickling cucumbers, and some in sweet corn as well. And I'll start today by just kind of thinking about this concept of soil-borne disease and the organisms that cause these diseases. In the examples I'm going to give today, we're going to kind of focus on three different groups. So we have fungal organisms, and these are causing fungal diseases. And then we also have fungal-like diseases. These are groups as oomycetes and include things like Pythium and Phytophthora. And the reason I want to take a minute to kind of think about separating these two out is that a lot of times they get lumped together. They both get controlled by products that are called fungicides. But these oomycetes like Pythium and Phytophthora, they have a different composition. So they actually have cellulose in their cell wall. And so the things that are going to work and target against Pythium and Phytophthora may not have activity on our traditional fungi and vice versa. So it's important when thinking about managing these of what you're trying to target, so you're making sure to select the correct product. We also have nematodes, which are microscopic worms. There's all sorts of nematodes out there, the animal and human versions. If you give your pet a heartworm pill, that's kind of the animal version. These plant ones are out there, and they have these pointy noses called stylets. It's basically like a needle that they'll poke into the base of roots and suck out nutrients. And so a lot of times the symptoms we see there might be stunting or yellowing. And sometimes the symptoms aren't that obvious until you actually dig up and start looking at the root systems. So I'll begin today spending some time talking about root knot nematodes. There's all kinds of nematode species out there, and across our region, we have many around. But root knot is one that's very visually distinctive because they really leave a calling card. When you dig up a plant and you see all these knobby nodulations on there, it's a pretty good indicator that you have root knot nematode. We'll take a minute just to kind of orient how this organism is working. So there's different types of root knot, and around here we mostly have southern root knot. The name of that one is Meloidogyne incognita. And again, it's kind of important to think about these different species because when we think of host resistance or other management, sometimes something might manage southern root knot, but may not manage northern or some of the other ones that are around there. These nematodes will survive in the soil as eggs, and they can survive for a really long time, which is why they become so challenging. And, it really lets them accumulate in soils kind of going unnoticed. Within this egg, it'll make the first juvenile, and eventually this will hatch out. And this is what's called a J2. And these juveniles or J2s are the ones that are going to move through the soil and actually find those root tips. So they'll find roots and then they begin feeding. And in the case of root knot nematodes, this feeding site that they make, they basically kind of cause things in the cell to go haywire, and they start forming these giant cells. So there's a lot of cell division that's happening. So all of a sudden, you start seeing this swell and get galling. And from there, that's where it's going to set up camp, continue to feed. These galls will expand, and then we may see symptom development and yield decline. And within each of these, the females will then lay eggs, and any given nematode has the capability to produce as many as 1,000 or more eggs in a single generation. And these nematodes complete a generation anywhere from like 3 to 4 weeks. So when we think about the course of a summer, these nematodes are able to have multiple generations And that's how these populations can really blow up quite quickly. So when you're out looking for root knot, some things you can look for, a lot of times nematodes tend to be patchy. It's usually not until they're pretty widespread that you see like uniformity across a field, but a lot of times there'll be hot spots. This is something that's in the soil. So anytime we're moving soil or equipment, it's slowly spreading them throughout the season. And you may see stunting, especially, in some of our bean crops and things. That stunting is really tricky because it can get confused with other things. It may not be that noticeable until you really have high populations. And same with some of that leaf yellowing and chlorosis. There's many factors that could give those symptoms. Another thing with root knot that we start to see is a lot of times midday wilting. When these roots get all galled up, they're not able to translate. I'm not sure why the PowerPoint keeps flickering, but anyways, when they get some of this midday wilting going on, they're not able to uptake water right. A lot of times we'll then end up seeing some leaf drop. And there's been some interesting work so far in tomatoes that actually show that when plants are galling, they sometimes release more ethylene, and it's thought that this may contribute. So there's sort of the water uptake part that's causing leaf drop, but there's also some things going on with ethylene release when you have this extreme galling. And when we dig these plants up, like again, the most obvious sign is seeing these gallings on the root, and a lot of times this will lead to some yield loss. I have this aerial picture in here just showing one of the fields where I had some nematode work set up this year. And you can kind of see where there's this darker green patch where there weren't as many nematodes versus this back of the field. And again, just kind of showing that, even in this field scenario where I'm trying to create an even distribution of nematodes, they do prefer and often tend to be patchy when you're out looking for them. Thinking about nematode populations is important because if you start the season with a lower level of nematodes, you tend to have like a little bit more tolerance within the crop, and you may not see as big of a hit on yield. But if you start the season already with really high populations, and then those are going through multiple life cycles, that's where we start to see some yield loss come to happen. So one way to kind of get a handle on knowing if you have root knot in your field is to scout. So you can do this in season by digging up plants and checking for galling. And this lets you know they're there, but it doesn't really help quantify. So in order to quantify and kind of get an idea of how many nematodes are actually out there, that's when we start to do soil samples. Soil sampling for root knot nematode, I typically recommend to do towards the fall. You want to do it while the crop is still in the field, nematodes are active, but around fall time, August, September into early fall is when nematodes are really active, and a lot of those J2s are out in the soil, and we can get a better quantification. You want to aim to take soil samples that are about 6 to 10 inches deep, and you want to take a whole bunch of samples and mix them together. You could dump these into a bucket and then ultimately put them into a bag that you'll send off. In field crops, we typically take a soil sample to represent every 20 acres or so, but in veggie crops, since a lot of times these are higher value crops, you may have smaller acreage and different rotation patterns. It's usually recommended more to try to aim one sample for every 5 to 10 acres. And again, recommending to sample towards the end of the season before the crop is destructed. In theory, you can sample nematodes at any time in the year, but that's just sort of a window to catch things when they're most active. And there's 2 types of sampling that you can head out and try to collect. One of those is predictive. So if you're just interested in knowing, how are nematodes distributed in the field? Do I have nematodes? This would be a predictive sample. And here you would just want to go out in the field and kind of zigzag through, make an M shape, a W shape, anything that's just giving you an kind of a swath across the field, and you'll mix these up, send them in your one bag. There's also diagnostic sampling. And so if you have a particular spot where yields always are bad and you know something weird is happening there, then you can sample all of your 10 to 15 samples just in that hot spot. And then you'll want to collect also in a nearby area that has some healthy plants, and that'll give you a comparison point to see what's going on in that zone. For sending off samples, there's a couple nematode labs on the East Coast. I tend to work mostly through the NCDA. They have a really active nematode lab there and a really quick, usually at returning samples. There's also a nematode lab at Virginia Tech. That one with COVID, there have been a few times where the lab isn't open, so just check to make sure they are open. But I think as far as I know, everything's back up and running there. And I will give a plug, if you have any veggie fields that you're rotating with corn or soybeans, my program does have some ongoing projects where we're looking at nematode sampling within soybean and starting this year in corn. And happy to tie this into some of the veggie production fields as well. So if you're interested in joining, I'll have my contact info here at the end, and feel free to send an email or give me a call if you have some fields where you are worried about nematodes. So when you send these samples off, you typically get a table that comes back and it lets you know, what type of nematodes you have, what populations they're at. And this little snapshot that I put in here was some fields that I had this year where they were particularly high nematode counts. And it'll give some recommendations here that you may want to consider. And some of the things that kind of are within our control to try to manage nematodes are thinking about crop rotation, host resistance, and chemical management. With nematodes, it's important to keep in mind that this sampling and the management piece is really going to happen before planting or after harvest. Unfortunately, once we're in the middle of the season, if nematodes start damaging the crop, there's not a whole lot you can do within a season to manage anything. Root knot nematode is a tricky one because it does have a really wide host range. So crop rotation is somewhat limited. In most cases, pretty much everything can be a host for root knot, but there's some plants that are worse hosts. So they may not allow the plant to, or the nematode to reproduce quite as much. And some of these that work in that vein are small grains, sorghum, and alfalfa. So the root knot can live on them, but we typically don't see reproduction at the level that we do on most veggie crops. So these are some options to integrate in, but unfortunately not a whole lot on this list that's helpful with root knot because it just is a host on so many of our veggie crops. Another aspect that we can try to target is host resistance. And as Emily mentioned earlier, she has a few lines in her screening where we're seeing some benefits. These are examples of some where there's both reduced galling, and then when we extract the eggs out, there's less eggs. So this is hopefully helping from two standpoints of having a healthier plant, but also limiting the amount of nematode reproduction that's happening. And we'll be partnering to do some in-field trials this season where we're looking at a couple of these lines that have been screened in some of Emily's preliminary trials and look promising. And then trying these in combination with a few of our chemical management options as well. On the vein of chemical management, I want to take a few minutes to talk about nematicides and how they work. And it's interesting because we pretty much call everything that has some activity on nematodes a nematicide, but most of our nematicides are truly more of a nemastat. And what this means is that kind of the old chemistries, the old fumigants that used to be widely used, those were true nematicides where it was when it was coming in contact with that nematode, it was affecting the nervous system and pretty much killing it outright. Unfortunately, we happen to have nervous systems too. So a lot of those old chemistries that worked really well had a lot of negative effects for human health and environmental health. So we don't have those options anymore. And as they've created safer products that are more targeted specifically just for the nematodes and insects, the trade-off has been that a lot of these are now nemastats. So what that means is they are products that will reduce movement. They may reduce the amount of mating or the distance the nematode can travel, how well it's able to make that feeding site established with the root. And it may also delay egg hatch and molting. So instead of being death outright from the chemical, it's more of a result of the nematode being disoriented and starving because it's not eating or making those connections of feeding sites. So the key here is that if exposure to some of these nematodes is shorter or at lower concentrations, these effects can be reversible. So just because we're putting a chemical treatment out, it doesn't mean that you're necessarily going to be wiping out the whole nematode population. So again, we have to kind of think about nematode management as an integration of a lot of different parts. And we have a few chemical options, but unfortunately, none of them are something that we can rely solely on. So you really do have to think about that host resistance piece when it's available and the rotation piece to really try to drop these populations. So within the chemical world of nematicides, we have fumigants and non-fumigants. And so these fumigant nematicides are often formulated as liquids that are going to vaporize, move through the open air spaces within the soil. And then we have non-fumigants, and I'm going to spend some time talking about some of these that we have. And these may include things like mocap, bidate, nimids, and counter. And these ones are often in granulars or liquids, and they're going to move through the soil through percolation of water. And so a lot of times these get classified as contact or systemic. So in some cases, they're contacting the nematode in the soil. In other cases, they're actually moving into the plant, and the nematode's making contact by feeding on the root of these plants. So within pickling cucumber, there's currently 3 main non-fumigant options that we have. The first is Vidate. And so this one is a carbamate insecticide and again, nematicide, but it really more is on that nemastat. What it's doing is basically paralyzing the nematode. So it's not moving. And because of that, we then don't get the reproduction and some of the effects there. Vidate is one that's both soil systemic and foliar translaminar. So it's able to move from the soil into the roots. And then if it's on the leaves, it's able to move down as well. And what it will do is then have some concentrations that build within new root tissue. And that's the site where the nematodes are feeding. So they'll feed on these roots, basically become paralyzed. And then we hopefully see throughout the season, lower end of season population, hopefully less damage and less galling, which leads to better yields. Vellum prime is another option that's out there. This one is a fungicide that also has some nematicide activities. It's again systemic through plant tissue and basically working by immobilizing those nematodes. So again, they kind of get disoriented, can't really move and make those feeding sites. And we also have nimids. And this one, again, kind of targets that acetylcholinesterase activity. So it limits motility and egg hatching, which leads to an overall decline in survival. In this slide, I have some data from some work out of Georgia. And in this, they were looking at some of these products at different inoculation densities of nematodes. So they were putting nematodes into these plots at either 1,000, 5,000, 10,000, or 20,000. And what they were looking for here, they are showing the percent reduction in galling and the percent reduction in final populations. And then we see that overall yield increase compared to the control plots where they had these nematodes added but no products. So in this trial, they were looking at Vidate, Vellum prime, and nimids, which I just talked about. And they also had a product in here that is called Celebro. And this is one that is not yet labeled. There's some things moving through IR4 right now, and hopefully this will be another option in the toolbox in years to come. But this is a new nematicide, and it still has an unknown mode of action, but again is targeting that mobility and infectivity of these nematodes. And so you can take a look through here. In general, the Vellum prime and the nimids, along with this new Celebro, had the most consistent reduction in galling and final populations. And also we're seeing some returns on yield increase as well. If we switch over into lima beans and snap beans, the product that we have labeled there is mocap, either as the 15G or EC. And this one is another one of the organophosphate insecticides. And this one, it's important to consider that it's nematicidal when it's at those higher rates. When it's at the low rates, we really only see the insecticidal activity. And this one is going to temporarily parasitize and paralyze those nematodes. But this one is contact activity only, not systemic. This one is a little bit less mobile and it can be highly bound to organic matter. So because of this, it is important to get a uniform soil distribution. And because this one's contact only, as you know, if we have a lot of rainfall, we have things where there's high moisture environments, it may leave the system sooner since it's not getting as much systemic activity into the plant. We'll wrap up with chemical options thinking about sweet corn. And again, we have that mocap that we just introduced, and another option here is counter. And this one is systemic activity, and again, targeting the acetylcholinesterase. So there, you can see kind of a theme in a lot of these new products that they're not truly killing the nematode, but they are targeting to paralyze and hopefully keep nematodes from eating and then killing them because of starvation. All right, so I'll take just a few moments if anybody has any questions on nematodes. We're going to switch gear into talking about some of our fungal and oomycete issues next. But any questions about nematodes? All right, so there's a question here about planting with rapeseed as an organic way to kill them. And this is an option, but the key is you have to incorporate into the soil. As with everything with nematodes, you know, we still haven't really found that silver bullet. Most of the data shows that if you incorporate in, you can get some of that fumigant activity, but it's still not going to be kind of like the silver bullet that solves everything, but that is an option. But the key is just the incorporation piece so that those are dispersed through the soil. If you just are planting it and then planting in, you don't get the effect. You really have to get that incorporated into the soil. So the next piece that we'll talk about is kind of early season and when we're trying to get good stands and good emergence. A common problem that we have is damping off, and this can be both pre or post. So in this video showing, you can see some little seedlings trying to come up, and some of them are dying in the soil before they even make it out, and we call this pre-emergence damping off. And then if it makes it out of the ground and then we have death, we call that post-emergence. And a lot of times damping off just gets lumped together. You know, the plant dies, it gets called damping off. But what's happening is that there's actually multiple types of organisms that can cause damping off. This could be Fusarium species from Rhizoctonia, Phytophthora, or Pythium. And it's important to think about which organism so that you can make the right approach to managing it. With Rhizoctonia solani, this one is a fungal pathogen, and there's lots of subgroups within this Rhizoctonia solani, and they get sorted out into these things called anastomosis groups, and a lot of times these end up being somewhat host specific. So this is a list just showing some of the anastomosis groups that are out there, and I've put in bold some of the ones that we see within corn, within bean, and some of our veggie production. This AG4 is one that I'll touch on in just a second. With Rhizoctonia, there's multiple types of infections. So we can have those root and stem rots that may be the damping off. It could be root rot or crown rot that happens later in the season. And then occasionally we also can get aerial symptoms, which are a web blight. One disease that we've been tracking in Lima beans is from that Rhizoctonia AG4. And this is an interesting one because what we end up getting is actually not really any symptoms on the plant itself, but within the pod, we'll get beans that turn brown. And so far, we've kind of just been referring to this as brown bean, but we get this discoloration, and it seems to be worse in wet conditions and if those pods have made contact with the soil. So this is something that we'll be continuing to investigate in 2021, basically trying to see if these are if this pathogen is present early in the season or if we can really only get these once beans are mature. And we'll continue to do some sampling as we have our root knot trial and some beans out there to observe this coming season. All right, so we have Rhizoctonia, which is a fungus, and then we also have Pythium, which is an oomycete. Pythium is often favored by wet conditions. Historically, we see a lot of Pythium in lower areas of the field where water might pool a little bit more. And in the case of sweet corn, this is one that we usually have worse issues with in early plantings, especially if those temperatures are hovering around 55 for soil temp or a little lower. When we have these early plantings that have slower germination, the seed is in the ground longer. There's just a higher chance for it to come in contact with the pathogen and then see these symptoms. As the season goes on and germination happens faster, there are actually, what we're finding is multiple species of Pythium out there, and some are favored by cool weather and some by warm. So just because it gets warm doesn't mean you're totally free from worry from Pythium, but it is often favored by those early plantings. And I have just like a snapshot here of some of the seed treatment products that we might think about for sweet corn, and just noting what they're active on. So it's important to note, again, if you have damping off issues of sorting out if you think it's a Pythium, a Fusarium, a Rhizoctonia, different products are going to have different types of activity. So our products that have metalaxyl or mefenoxam as an active ingredient are going to have Pythium activity. And then things like azoxystrobin or fludioxonil, those are ones that are going to have activity on true fungi like Rhizoctonia solani. Then there's products out there like Thyrium. This one has a very vague label that just sort of covers all damping off species. For optimal Pythium activity, it's usually out of these metalaxyl and mefenoxam products. But we do get some fairly broad spectrum there. I've also included this table. Now this comes from soybeans, but again, just trying to reiterate, this is a soybean table where we rate E as excellent, and I've boxed in, again, just considering these products that are excellent against Pythium and Phytophthora have no activity against Rhizoctonia. So when you're in, other crops, if you're growing melons or, other similar crops, we see a lot of damping off, and just sorting out which one you have is important so that you can select the right product. I have just sort of a snapshot here of just some other interesting things that I saw throughout this season. Today I spent time talking about Soilborne, but there's certainly plenty of foliar disease that's out there. And in these two pictures, this one here and the one on the end, these are highlighting some Fusarium samples that I got this season that were some new species. And I will put a plug in that we do have a new person at our diagnostic clinic. So if you haven't had a chance to meet Jill yet, she started right in mid-pandemic. Nancy Gregory retired last year. So I'm down here at Carville, but Jill's up at main campus as well. And the diagnostic lab is open. And there's also a new Instagram account from the plant clinic, and lots of activity there as Jill is getting settled. And I'll take a moment. I always like to give a plug if you are submitting a diagnostic sample, some things to consider. And so, virtually, if you want to take a minute just to look at this picture and think about if you see Wolverine or do you see two Batmen. And I give this picture just again to think that what you see might not be what others see. So when you're submitting some diagnostic samples, try to give context. If it's only a crumpled dead leaf, it's harder to piece the story together and figure out what's going on. And then as you're taking pictures, it's always great if you have something that's zoomed in, but then maybe further out to give context of what's happening across the field. And if you are collecting samples, remember to try to put them in a Ziploc bag or in a cooler. Don't throw them up on the top of the dashboard so that they get dried out and crispy in the sun. It makes it harder to figure out what's going on there. So just a few notes on diagnostics. And I would like to just acknowledge my lab group. I have 4 master's students right now that did a great job this season of trying to make the best of our weird work situation and still have a very active field season. And hopefully I'll see some of you next week in the ag session where we'll be sharing some results from our soybean, corn, and wheat work, and acknowledge some of our funding sources here. And then with any remaining time, I'll be happy to take questions.